Thank you, Keith, and thank you for the invitation. It's been a great meeting. I wanted to talk about something slightly different. So we've been talking about synthetic biology and engineering in the context of what happens inside cells, rearranging metabolic pathways, etc. And I want to talk, or really the focus of this talk is what happens between cells and how you might re-engineer form or the arrangement of cells in a living plant. And I'm starting out here, this is obviously a coffee cup. There's something like 200 billion paper cups made each year on the planet, and they're all made the same way essentially. It's a template-driven process. Natural resources are harvested, directed to a factory, processed, and then stamped out, template-like, and then assembled into a paper cup, of course. And in biological systems, the process is extremely different, of course. And this is appropriately from Bristol. This is from the BBC Natural History Unit based here. And what we're looking at here is a time-lapse image. And this shows the leaf of this particular species with a stolon emerging from the leaf tip. And as this time-lapse progresses, you'll see this little nub of tissue here will develop, it will enlarge. And you'll see here it's enlarging into this, it's actually a hollow structure, very much like a coffee cup. And it has, in fact, a lid up here, which is forming. And as you see it growing, you have the coordinated activity of many millions of cells enlarging, dividing, of course, enlarging, differentiating to form this very precise architecture. And here it is. It's resting on the ground by virtue of this elongated stalk. And here it is. It's about to open up. And of course, it's a pitcher plant. It's a Nepenthes species. And you have what looks like, to all intents and purposes, like a coffee cup. And when you look closer, it looks even more like a coffee cup. If you think of our Starbucks cup up here with the rounded lip and the polyethylene lining, here's a pitcher plant. Here's the rounded lip. Here's the wax epicuticular lining here, which where unfortunate insects may slip in and not be allowed egress from the container. So this process produces analogous structures, but by a very different process. And so in the living case, rather than it being template driven, it's driven, as you might imagine, from the bottom up by virtue of coordination of cell activities. And it's this process which is quite, in a way, mystical because of the emergent properties that embody this process. And so if we think of plants, well, here's the growing point of a particular plant species where you've got the meristematic cells which contain the stem cells, which retain the properties to divide and to deliver cells to the growing organs around it, which in turn expand and differentiate to produce the adult structures. And so the process of growth involves this spatially partitioned and temporally organized process where you produce the kind of thing that we saw in that time-lapse process. But of course it's driven by local cell activity. So each one of these cells here, which you can make out in cross-section, is undergoing its own particular activities, encoded essentially by interactions by its immediate neighbors. And so you have this large-scale structure, like a pitcher plant, which is undergoing this organized, quite precise process by virtue of local interactions where individual cells know nothing of the master plan. There is no master plan. All they can tell essentially is what's happening on around them. And so each cell is responding according to a common genetic code that they're all sharing and responding in a coordinate fashion, which is quite magical really. And this process of cellular elongation and division takes place within an immobilizing framework. So unlike animal systems, which are free to migrate and generally undergo apoptosis when they're not in the wrong place at the wrong time, plants by necessity, have a very flexible arrangement. So cells, as they grow, get to a point where they want to divide, and the two daughter cells here are essentially separated by a new wall which is built in place. It's like subdividing your flat, that you're building a wall within the existing architecture, and that this and all the surrounding cells, which are not shown in this diagram, are immobilized by this shared composite cell wall structure. And you can see the two layers here, each of which is contributed from adjacent cells. So these two daughter cells, shown here after division, are locked in place. They and their descendants will share this boundary for the rest of the life of the plant. And this process of immobilization at the microscopic scale, of course, as well as the macroscopic scale, impinges constraints on development such that you can characterize the pitcher plant development, or at least one portion of it, in this rather highly schematized version, where you have individual cells within the body of the plant which are receiving their information from their neighbors, and only from their immediate neighbors essentially. And this process of information exchange is a two-way process, and that this determines the fate of individual cells in these developmental processes. And in this case, in the schematic version, we've got this cell is now being programmed to divide. And so these two daughter cells are now by definition in different places inside that organism, receiving different sets of information, and of course contributing information back to their neighbours, which can be differentiated. So you have this bootstrapping process which links gene expression and cell properties to the local microanatomy of the plant. And this process will take place for a single cell in these dynamic systems, but of course it takes place for all of the cells. So you have this network of interactions which binds these structures together by local interactions. Now, the question is, how might one organize such a distributed process? And clearly, systems have evolved which have very precise and remarkable structures in plants and other organisms, of course. But the question is, might one rationally control such a distributed process? And that's really, that encapsulates our interests at the moment, which is how can one do that? So I want to start with our first attempts to try and rejig gene expression in plants. And these rely on small transcriptional control units, if you like, based on foreign transcription activators. And they're shown schematically in this panel here. There's two of them shown here in the top, which is based on a yeast transcription activator, GAL4. Another one down here, another yeast transcription activator, HAP1, which are modified for expression in plants and which can be used to tap into various promoters or enhancers to drive different patterns of expression inside plants. And these are hooked up to fluorescent proteins, either an ER localized green fluorescent protein in this case, or a nuclear cyan fluorescent protein in this case. And so you can develop individual lines of plants which turn on genes or can turn on genes at particular times and places during development, which can be really very specific. The two systems are orthogonal, so you can put them together. And this is one example here where we've got an Arabidopsis plant, a root here, where these cells at the very tip on the outside of the plant root are expressing this HAP1 protein, therefore expressing the cyan fluorescent protein. And up here you've got another set of genes which are expressing this green fluorescent protein. And they're essentially little pathways which are orthogonal to plant development. So in this case here, they're just driving marker genes. They're not linked informationally to the rest of the plant's activities. They're just being driven in an output, essentially, by virtue of enhancers which are next door to them. But what happens if you now start to hook it up to something which changes the plant architecture? So you can do that by using, for example, a protein which is from a DNA virus, in this case, beet curly-top virus. And there's a protein there which is, interacts with retinoblastoma protein. So it's a protein which triggers ectopic cell divisions. So you can take one of these lines, this is an example here, which is normally expressed in this outer layer of cells. This is a projection image of the very tip of a root from a mature Arabidopsis embryo. So it's just these little single layer of root cap cells at the very tip of the embryo, very selective, specific set of cells which are expressing this marker gene. And it's got the yeast transcription activator there driven from this particular line. So you can use that marker gene or that transcription activator to trigger another gene. And in this case, we've triggered ectopic cell divisions by expressing this viral protein. And so you can see here you've got this change in the morphology of the plant. You've got altered patterns of fluorescence and the shape, the number of divisions that take place in this outer layer have changed. And we've chose here specifically a line that's expressed in these outer layers. However, if you look inside the plant using imaging techniques, you can see this is the wild type situation here, which is just this driver line. And it's these white cells on the outside that have been affected normally. And in the case of the expression of the cell division protein, you've got this ectopic divisions which contribute to a larger number of cells in that layer, which is cool. That's probably the primary intended effect, if you like. But what's happened inside here, in the body of the meristem, is there's also changes. For example, these yellow and green cells all derive from a common initial. Just as a diagnostic here, you can see this common initial has also is proliferating without undergoing the normal periclinal division. So you see effects not just due to expression in the targeted tissue, but secondary effects. And that's consistent with the idea that all of these cells are hooked up to each other. They're all listening to each other. And if you change one cell on the outside, one cell type on the outside, then that information propagates across the system. And meristems are characterized by this process, and that contributes to the robustness and the self-organizing properties of these systems. However, if you want to rationally rejig or reorganize properties in here, you have something of a problem, or more of a problem. And so what we are aiming or hoping to do is to generate systems which are not like, you can imagine there's these 3 generations of types of gene expression in a very general sense. And if you imagine the first generation of gene expression approaches involve just promoter gene fusions, taking a promoter, an ectopic promoter, and turning your gene on in different places. The second generation is what we've just seen, using a transcription activator to form a small pathway so you can get coordinate division, coordinate expression rather. And these are essentially analogous, except here you can switch or regulate things in a coordinate fashion. But what we really need are systems that will trigger gene expression where that pattern of gene expression is then autonomous. You can trigger something which, once triggered, has its own internal forces which control gene expression. And these borrow from the kind of ideas that Alan Turing expressed in the early 50s. And just to remind you what just before he died, some of the ideas that he propagated were the idea that you could generate order in a spatially organized system by very simple processes, that is noise and diffusion or transport, and that these are enough to build order. And this is sort of paraphrasing his initial work, which is that if you have a system where you have an activator of any type which feeds back on itself, so you have a positive feedback loop, if you have a noisy system, then there are going to be some elements which are above average, which because of this positive feedback will tend to increase. And at the same time, this activation can spread. So as you activate it, it forms a domain, a small microdomain. But at the same time as this process is happening, this positive feedback, you also trigger negative feedback. And this process of production of some kind of inhibitor or poison, as he put it, involves triggered production of an inhibitor, which has faster transport or diffusion properties. So this will be produced by the same process and will self-limit this process. And so this has the property within a noisy system of creating ordered domains. And so you have this positive feedback loop with this lateral inhibition. And this is seen in many biological systems in various themes, but in its very simplest form can produce these patchy stripe-like processes or dotted-like processes, seen, for example, many pelts of animal systems due to these, or presumed to be due to these kind of processes, early embryogenesis, and tropical fish which have these. As they grow, the stripy patterns change as they change the spatial dimensions. And that's the simple case. This shows, actually, this is, I think, a really nice illustration of it. This is from Jonathan McCabe, who works in Canberra, and he's actually a computational artist. And so he's taken Turing systems, and rather than having a single simple Turing system, he has overlaid Turing systems which have different spatial dimensions. And so these can form these highly randomized processes which form these rather organic pictures, because you've got self-organization. We started from this noisy field, and it builds order by virtue of these self-organizing processes, breaking symmetry and creating these self-organizing domains. And these refine, and in this case they change actually because they're not constrained in that way. But the parameters can be used to generate order and to modify in a rather organic fashion. Hence the name of this piece is called Bone Music, and it has these organic features. So these have extremely interesting properties if you want to control gene expression in a way that you can trigger something and then have some kind of self-reinforcing boundary, or to create a process which will then be autonomous. And the kind of approaches that we've been developing over the last few years involve, or increasingly involve, this kind of cycle of development where we have obviously DNA elements, and adopting some of the ideas that have been popularized by Tom Knight and others. The idea of using modular parts makes this thing more general, and you'll see why in a minute, that you would want to use modular parts to experiment with the creation of these kind of circuits, to produce DNA elements that can be put into particular chassis. We're interested in plants, but we use microbes as a tool for this. And then using increasingly imaging techniques, particularly quantitative imaging techniques, as a way of not just visualizing the phenotypes of DNA encoded processes, particularly cellular processes, but also to obtain quantitative data and to feed these into software models. And the reason why this cycle is so important for us is that we're dealing here with essentially emergent properties, but also nonlinear properties. And the relationship between the DNA circuits themselves and their cellular phenotype is not self-evident, and that software models are the only way of really handling the kind of parameters that get involved in these kind of processes. So, actually, before I go on, I've color-coded these different processes, and I just want to quickly run through the kind of approaches that we're using. So we have an instance of the JBay registry that we use in Cambridge now, and it has very nice facilities for storage of DNA, for display of DNA. There are a series of plugins which are overlaid on the database. So for example, you can keep sequence traces. Everything's automatic, so you just feed in the sequence trace files, it maps it onto the sequence, highlights any potential errors or misalignments as you get at the ends of sequence runs, for example. It allows you to have access to different plugins for assembly and for vector assembly. And this just sort of describes these smart registries that you've got this, not just the storage of the DNAs themselves, but also these series of plugins which allow automated assembly and oligo ordering, etc. And these are the kind of, it's an extensible registry and database which allows plugin modules as an API which allows programmatic access. And this is, I think, not just a useful thing, but something which can be extended and is an extremely useful development in the field. Oh, I should say that Nathan Hilson and Tim Ham are the key developers at JBay, but it's an open source project and they're very keen to encourage collaboration. And so using these kind of accessory tools, and we've got one in Cambridge called Gibthon, which allows automated Gibson assembly. It allows you to use these, integrate these into a workflow. This is roughly what we're using here. We seem to be losing a little on our slide here, but so we've got these small parts that can be strung together with Gibson assembly, and we use both Bacillus subtilis as one vector and also agrobacterium-based plasmids to generate these systems for chromosomal integration in both the microbial and the plant systems. And using these kind of processes, you can collect and assemble these different parts. We've got pigments here and luminescence systems. These are from iGEM projects, and this is like these. So Ben and Theo here are being photographed in the light from their own experiment. So these are from highly luminescent microbes that they've generated. And also for the kind of systems that we need to build these Turing-like systems, which involve cell-cell communication. And so these are, first of all, homoserine lactone-based systems, but increasingly peptide-based systems, which allow direct linkage between transcription in this cell and that cell. So you've got these simple microbially derived systems for communication between cells, which you can test and retain as parts in this process. So if you've got parts, then the question is how you reprogram systems. And perhaps counterintuitively, we've been using microbes as analogues for plant cells. And we use high-resolution imaging techniques, a lot really. And this shows some of these microcolonies which are growing under a cover slip. And this is Bacillus subtilis. This is a colony which would look, at this point, this is the 50 micron scale bar here, so we're talking about a rather small colony. But you can make out individual cells here, of course, in these processes. So where we're trying to build systems where we can visualize communication between cells, this is really important. We've developed these vectors, and this is James Brown's work in the lab mainly, where you can use ratiometric imaging to visualize not just essentially whether something's expressed within cells, but quantify patterns of gene expression and promoter performance in these microbial systems using, essentially algorithms which take into account growth and stability of the fluorescent proteins and transcription rates and turnover rates. And these give very nice linear traces, which is a complement to Jason Kelly's work using a single GFP production. And as I mentioned, imaging, so this section is all about looking at living systems, and imaging forms a major part of this whole process, where this shows a young Arabidopsis embryo, and the ability to use confocal imaging and reconstruction allows you to get a three-dimensional view of plant tissues, but also to build three-dimensional images. Both, in this case, we're looking at fixed tissue with some staining procedures we've worked in the lab, which are essentially reiteration of very old-fashioned micro technique adapted for modern confocal microscopes, and using modern image processing techniques to build three-dimensional representations. So you can virtually dissect three-dimensional architectures, for example. And with live tissue, you can go even further. So you can use now different fluorescent proteins where you can, say, for example, label nuclei in this case with one color, and using a special membrane label which doesn't internalize, doesn't endoscytize. It gives you two labels which can be hooked up to image processing techniques. So you can use the red channel to identify cells. So you can count cells essentially using particle analysis technique, and that provides a seed for a balloon segmentation algorithm, which then uses the green channel to essentially segment cells within a two-dimensional slice or a three-dimensional tissue. And so you can get accurate and robust creation of segmented cell patterns inside tissues. And this is the use of these two live markers, which I think is novel, allows this robustness because the balloon segmentation can have contact detection essentially between adjacent cells. And if you know anything, if you've done segmentation, you know that poor quality data leads to bleed through and causes all kinds of pain. And this is a very robust process which is useful for live tissue imaging. And of course, you can use this in intact tissues. Here we're looking at the stem of a young Arabidopsis seedling, the two hypocotyls, and the central portion of the apical meristem. And here we've got not just one nuclear marker, but two. In this case, we've got a cyan marker in the membrane, but this has got both red and green fluorescent markers in the nucleus. And so you can use this to generate a ratiometric characterization across the cell, across cell boundaries essentially, or across a tissue. And so you can quantify the relative expression of a single or two sets of genes in a single cell and measure the green and red fluorescence from each one. And of course they're normalized and they're in the same position inside the cell. So they're all competing, these are histone fusions, so they're competing for the same site on the chromatin. And so you can generate these cartoon-like views of plant tissues using this combination of robust segmentation and ratiometric imaging to give quantitative views of not just gene expression at any one point, but also differentials. So if you're looking at a process that changes over time, you can see things which are not evident by eye, of course. So here we've got, for example, strain rates. So you can use the segmentation process to look at the changes in cell shape over time and map that onto these tissues. And here we've just got two, and these are looking at cytokinin treatment of a Arabidopsis root. And so we think this is something which has legs. I think this is the kind of thing that is very useful for systems analysis of dynamic tissues and feasibly could be built into a microscope for live imaging. So we've looked at the parts and the biological system. The question is software models. And we have a collaboration with Andrew Phillips' group at Microsoft Research on the use of software to compile circuits. And I won't belabor this too much because you've heard quite a bit about this already. The idea that you can put together parts to create local circuits which can then be fed straight into a model of, and the Andrew's group uses a pi calculus-based system called GEC, which has incorporates some of the stochasticity of models, but with the economy of differential equation models. But what you can also do, and this is our particular interest, is to plug these kind of genetic programs into a cellular automata models, which include not just the genetics that might happen inside a cell, but also include the spatial and temporal aspects of cells interacting in a process. And so we have in these models, not just genetic programs, which of course are identical within each cell, but also models which include the description of the mechanics of the system, and also include algorithms for cell division and the like from cell biological studies. So the kind of things that you can do here are, for example, here we're looking at microbial cells dividing, and this is showing a junction between the two cells here. And there's another system here showing differential signaling between these microbial cells. Now these are cellular automata models. Each one of these cells really is an element in the computational model. And these are, we've spent the last year or so using GPU acceleration to generate systems that can handle 30,000 or so cells in very fast time. And if you're prepared to wait a little longer, you can go up to full colony size. And so the key thing here is that we want to build these morphogenetic systems that allow you to deal with large populations where you can integrate these kind of Turing-like processes. And so I'm not going to talk too much about the Turing processes. It's still essentially in process. But what I will talk about, given the nature of the meeting, is the plant work that we're doing. And so we mainly are working with Arabidopsis, but we've increasingly shifted to these simpler and simpler plant-like systems. And there's two I want to just briefly talk about. So this is the Arabidopsis root meristem. And by essentially any definition in terms of higher plants, it is simple. It's one of the attractions of Arabidopsis as a model system. But this is Collichete. So it's from a key genus, which is essentially quite pivotal between multicellular algae and the first parenchyma-like plant systems. And it's highly reduced or simple in architecture. So it forms these plate-like structures, a single cell layer flat, and grows by cell division only in the edges of these colonies. And they start out of these single motile spores, if you like, and they form these cellular systems that grow on immobilized surfaces. And over a series of experiments over the last couple of years, there's been, I think, two quite important papers, one from us and one from Jacques Demay's lab in Harvard, which have described the growth of these very simple systems where the developmental processes and morphological processes are laid out extremely starkly. And so in our experiments, we showed that the patterns of cell division in these processes are essentially constrained or correlated highly with the shape of the cells as these meristematic layers grow out. So that the process of cell division can be essentially described by a local cell autonomous response to changes in shape. So as these cells are growing, they change their shape and they're constrained by the physics of the interactions between the neighboring cells. But essentially it's the shape of the cell which describes which way they divide. And Jacques' lab, and Jacques is a proper biophysicist, has come along and generated a cytoskeletal-based model for how this might happen. So you have what the equivalent probably would be microtubules, and this harks back to Clive Lloyd's work at the John Innes maybe 20 years ago, showing that this could form a mechanism where you've got equalization of the energy of the system to create a way of calculating space and shape inside the cell and to determine the patterns of cell division. And this applies very nicely to Collichete as well. And so this process fed into now a plant equivalent of one of these cellular automata systems, where we've got individual cells which are communicating with each other, which give rise to these dynamic models, gives you a system, when you apply these rules, of modeling Collichete. So here's a live version of Collichete, or a time-lapse series of Collichete growth. And this shows a computational model. And some of the things that come with this model come for free. So for example, when real Collichete are disrupted, they can grow outwards, and they form this branched process here. When you take the model, which doesn't have any direct encoding of this process, the same kind of thing happens because you've changed the biophysical constraints on these cells in the very edge, the dividing cells on the edge of the colony. So they're unconstrained, they grow outwards, similar to what you find here. If you look at Collichete colonies that are growing together, if they collide, they constrain each other, so they form these characteristic shapes. Again, with these models, as they come together, they form these constrained shapes. So one of the things that's come out of this way of looking at cell division is, importantly, all the decisions are cell autonomous. So the cells here don't have to know anything about the greater shape of the structure or even really know what their neighbors are doing. They're just constrained or not. So it's basically physics, and it's transducing the physical constraints into patterns of cell division simply by calculating the size and shape of an individual cell. Essentially how all unicellular organisms do it already, which is, I think, quite an attractive aspect of the process. But, and just to finish really, the process of growth involves these patterning processes and then subsequent partitioning of those processes and then implementation of some kind of differentiation process, this last step here, if you like. And it's this differentiation process which is also quite important in different contexts. So for example, we know from study of essentially all multicellular organisms that development in genetics tells us that it's quite modular. If you make defects in floral architecture, this is obviously Drosophila patterning genes, segmented elements. This is looking at evolutionary relationships between related segmented insects, and is Antirrhinum down here. We know that you can take single mutations and cause quite large disruptions in these modular processes. And in order to generate a foot where your antenna is, it requires that there be some kind of slippage in a program, but everything downstream of that is still running appropriately. So there's some aspect of modularity and hierarchy which controls normal development in all processes. And the question is, if you want to have a system where you can control this process, in other words, what those cells will be and how they'll interact, you need to tap into this system. You need to be able to understand what the appropriate entry point for these decision-making processes is if you want to properly genetically perturb it. Otherwise, you end up with the first experiment I showed where you're misexpressing cell division and you've got unintended consequences. Whereas here, of course, you're changing things in a modular fashion. Now, we know in Arabidopsis you can do this. This shows a normal Arabidopsis leaf, and this is a leaf where you, Fernand in the lab has misexpressed VND6, which is a transcription regulator, which triggers xylogenesis. So every one of these cells on the epidermis is now turned into essentially a wood cell. So you can use this, and it's part of a collaboration with David Benjamin in New York, who's an architect and interested in this process as a way of creating shapes and creating wood-like structures. But this process is, in a natural system, is one part of a hierarchy. And you can essentially identify, from the DNA parts and genes and circuits that we tend to talk about, as you go up the hierarchy, you're talking about cells and tissues and organs and organisms and populations, that this process is linked in the normal system. And if you want to perturb this process, and VND6 presumably acts to trigger a genetic circuit, which then controls a whole framework of genes downstream of that. There'll be clusters of genes downstream of one of these decision-making processes. But in order to comfortably do this kind of work in the future in plants, you need to identify these points of entry, if you like. And for this, we need a simple plant test bed. And you'll see that through this talk, even Arabidopsis could be criticized as being too simple in a way. But we want even simpler systems to test drive these kind of things. And I just want to finish with a plug for this particular system here. It's a liverwort, which is right at the base of the plant lineage. So we have here the ancestral green algal, which is believed to give rise to green plants. And you see the branches giving rise eventually to modern angiosperms. But right at the base of this sister lineage here, you've got the liverworts, which are arguably the simplest processes. And this particular liverwort, which is Marcantia polymorpha, you've probably walked past it today. I know this is growing outside in cracks in pavements and what have you. It's a weed. It's quite a prolific little thing, but it's quite easy to pass by. It's only like so high. And it has this flat thalloid-like growth. It grows as a flat sheet and has a curious lifestyle where it will produce these little cup-like structures. And these are splash cups. A drop of water in here will distribute the kind of, you see these little propagules inside. This is one of the reasons why it's quite a pernicious weed, that you can, as they grow inside these splash cups, you've got cells on the outside of this layer in here, which will produce new organisms essentially by vegetative reproduction. And these will be liberated. So the things you see up here are little propagules or gemma, which are being liberated from this process. These gemma are quite accessible. You can put a microscope over the top of them, and you can section entirely through this is 3 orthogonal views through the same gemma. As they grow, they form these flat sheets, and they have these primitive but modular-like flat sheets. So each one of these jigsaw-like shapes is a module, a photosynthetic module, with an air pore at the top. So in cross-section, this is the air pore, this is the top surface, and they have these little photosynthetic chambers inside it, and they're just these repeating units. So they're very morphologically very simple. What's more, they're also genetically extremely simple. So here we've got Marcantia compared to Arabidopsis and other lower plants like Selaginella and the moss Fiscomatrella. And here we've got gene counts, the numbers of genes of particular families. And those of you familiar with plants and developmental systems will recognize auxin-regulated genes here, homodomain protein genes, etc. And you've got these families which in Arabidopsis are not too large, but in Marcantia are reduced either to solitary genes or small groups of genes in the case of auxin response genes. So you've got a highly simplified genome, which to us is extremely attractive. And what's more, the process of growth in Marcantia involves not just vegetative propagation, but sexual propagation. You've got male and female plants, which have, can produce motile sperm and have the archegonia in here. And you can do a simple cross between male and female and produce these, the diploid cycle is very short. The plant is haploid, like a yeast essentially. And they undergo mitosis and form these spore capsules, which are these little yellow lobes here. And each one of those contains about 500,000 spores. And so a single cross can produce millions of spores here. And what's more, they could be germinated on agar, and a week later they form a plant. So this is after a week of growth on a rich medium, and this is obviously a pipette tip for scale. But each one of these is a quite nicely formed small multicellular plant, which is in essence suitable for screening. So we are shifting all of our work essentially from Arabidopsis into this very simple testbed system. And I think it has a lot of prospects for synthetic biology type experiments. And I haven't mentioned that it's haploid. Homologous recombination gene replacement works in this system. And there are a number of other advantages as well. Anyway, I just want to finish with the last slide, which reprises the slide that Ray had in a way. So this is from John Dobley, of course. And this process of human selection to give rise to modern plants doesn't involve tinkering too much, or certainly not directly, with biochemical pathways, although there may be some difference in the biochemistry of a Peruvian tomato and a modern tomato, particularly in alkaloid content. But really, it's the major difference here is in the trajectory of development. It's the cellular interactions which underlie the development of this versus that are the key to that process. And I think there's some prospect, at least, for, in a way, the cellular aspects of development are a simplifying aspect, I would say. And I just want to finish with thanks to the people in the lab doing the microbial, the plant, and the computational work in the lab. And thank you for your attention.